mediating root growth in rice. And this is a work by my graduate student, Yijie Chen, and so I'm reporting the work on her behalf. The major effort in my laboratory is to discover the function of the so-called orphan stress proteins. So why we want to call this protein orphan stress proteins? Because omics approach, as we have heard many times in this conference, has allowed us to identify many genes and proteins whose expression is highly induced by various stresses. Yet the function of many of those stress-induced genes or proteins have not been really elucidated. So that's the reason we call them orphan stress proteins. While the omic approach is wonderful, but it's equally important that we understand the function of individual gene or individual protein. So the approach we've been following is very simple. We combine protein biochemistry, cell biology, and genetics, and we work on one gene or one protein at a time. So many years ago, we did something just like everybody else have been doing today. We did a very simple genome-wide identification of root-specific and also stress-inducible genes in rice. So by simple microarray analysis, we discovered many of those genes. And one of them stood out and caught our eyes, and we thought it's very interesting. So this gene is highly induced in roots when roots are under osmotic stress, soil stress, or cold stress, or treatment with ABA. As you could tell here, normally you don't see a whole lot, but under ABA treatment for a very short time, 2 to 3 hours, you see a tremendous induction of this gene. In the shoot, you don't see anything similar. And we verify the information quantitatively by real-time PCR. So on the left here, you have the real-time PCR result, and on the right, you have the same result, but only conducted with shoot tissue. So this is a root-specific, highly stressed and ABA-induced transcript. And very quickly, we know this transcript actually represents 4 different genes in rice genome. And there are 2 pairs of them. I'll tell you why they are called REPRP, because they are proline-rich proteins, and they follow certain kind of repetitive motif. So we call them REPRPs. And very quickly we know this is extraordinary because about 60% of the amino acid residues are proline. We have never seen anything like this. In the literature, there's nothing like this. They are proline-rich protein, but not 60% of all the amino acids. So there are 4 genes encoding 4 different proteins. So here is the alignments. And you can see there are lots of proline, and also oftentimes proline come in pairs. So they have the motif shown at the bottom of the slide. They come in as a PX1, PX2 motif. The X1 and X2 are usually highly charged amino acid, either glutamic acid or lysine. So you could have a motif like PEPK, proline followed by glutamic acid, followed by another proline, followed by lysine. And this could be repeated many, many times in these 2 different proteins. Although there are 4 genes, they come in pairs. And those pairs, the 2 components in the pair, are very, very similar. So at the protein level, we cannot tell the difference. So basically, we have a subfamily 1 on chromosome 5, and we have subfamily 2 on chromosome 7. And it's very difficult for us to differentiate 1.1 from 1.2 and differentiate from 2.2 from 2.1 at the protein level. So basically, we are working on two different proteins we call P1 and P2. And of course, at the protein level, the P1 and the P2 are also induced by stress, as shown by the Western blot here. But there's a difference, because we know the expected size of P1, P2, and the Western blot indicate the final form of protein is much bigger than the original predicted P1, P2, so indicating the protein is modified, actually heavily glycosylated. And the four different proteins expressed at different level, and so we are working on the two of them, P1 and the P2. And phylogenetic analysis indicate these REPRPs are unique in monocots such as rice. Although we have protein-rich protein in other plants such as Arabidopsis, but this kind of REPRPs are only in rice and other monocots. So as shown on this slide, so the shaded area represents REPRP, the others are the regular protein-rich proteins. So they are very unusual, and we already suspected that they were glycosylated. So indeed, if you scan for the presence of carbohydrate in the protein by the so-called PAS scan, and you see on the left, you have the protein gel. This is the PRP1. And on the right, I hope you can see on the screen, that is also PAS positive, so indicating this is a glycoprotein. The rest of the lanes are the positive control and the negative control. So this is a heavily protein-rich protein with carbohydrate attached to it. But there's only one end-linked glycosylation. And one end-linked glycosylation does not account for the molecular weight increase that we observe on the gel. So using a very sophisticated chemical analysis followed by mass spec analysis, we notice about half of the protein residues are converted to hydroxyproline. And among those hydroxyproline residues, many of them are glycosylated. Although we did not see the whole protein in the mass spec spectrum, from the partial spectrum that we could decipher, there are at least 8 O-linked glycans there. We don't know exactly how long they are, but we know for sure there are many O-linked glycans. We could determine the carbohydrate composition in those glycans, and these are the results. The control, this is so-called arabinogalactan protein that has been reported in the literature. We use a gum arabic as a control. Indeed, we see arabinose and the galactose. But in our P1, P2, the composition is different. In P1, we have arabinose and the glucose, although we do see some galactose. In P2, we see glucose and the xylose. We cannot even detect arabinose and the galactose. And so this is different from the traditional arabinogalactan protein, AGP. This is further demonstrated on the right. If this is an authentic AGP, it could be precipitated by a special agent called Yarith agent. So this is the positive control. But for our protein, they remain a soluble protein in the presence of Yarith reagent. So these are not typical AGP because they do not interact with Yarith agent, and also their carbohydrate composition is different. So this is a summary of all the hydroxyproline-rich glycoprotein that we know, including ours. So we have AGP. It has a lot of protein. It's heavily arabinose and the galactose. And we have extensins, and they are moderate glycosylated. And there are protein-rich proteins present in Arabidopsis. They are only lightly glycosylated. In comparison, our REPRPs are heavily glycosylated with galactose, arabinose, glucose, with a lot of different things. So that's different from the AGP. Probably the most striking observation we have made is the REPRP in rice actually is a lectin. It could bind to special oligosaccharides that are present in cell walls. So this is the important summary. We try the binding of REPRP with the different carbohydrates, different oligosaccharides, xylin, xyloglucan, glycan, and others. They don't bind. Only one thing binds, that's arabinogalactan. This is actually originally isolated from a cell wall. And we do the competition experiment. It turns out the P2 has the highest affinity toward arabinogalactan. P1 is much lower, and the traditional AGP does not bind very well. And also, we try different concentrations of arabinogalactan. Those are the hemicellulose available in the cell wall. So with the different concentrations of arabinogalactan, we see an increase in binding to the hemicellulose by PRP1 and also PRP2. As a control, GST or other proteins just does not do it. So let me repeat, this is very important. The rice REPRPs not only is a unique glycoprotein, but it's also a lectin. It binds to a particular hemicellulose in the cell wall. So now moving on to study the cell biology of this protein. So we do the localization of this protein both in stably expressed transgenic rice or intrinsically expressed protein in onion cell. In both cases, this protein is preferentially localized on the plasma membrane. In the onion cell, we could do plasmolysis. If you plasmolyze it, the green signal goes with the plasma membrane. But one thing important is, in addition to plasma membrane, you do see something else in the cytosol. We don't know exactly what something else in the cytosol contains this protein, but primarily this protein is associated with plasma membrane. And so from cell biology, we do the reverse genetics. As a control, we know ABA inhibits root elongation in rice. Yet promotes lateral root formation near root tips. So that's something already available in the literature. So we overexpress this REPRP and see what happens. To make a long story short, if you overexpress PRP2 in transgenic rice, it will mimic what ABA treatment is. So here's the control. And if you overexpress PRP2, and the root length decreases. And this is pretty much the same as if you treat the root with ABA. So once you treat the root with ABA, the control, the non-transgenic, becomes much shorter, which is the same as the P2 overexpression. Let me see whether I could advance the slide. Yes, okay. And if you do the reversal, if you RNAi, since there are two sets of genes, we do the double RNAi because single RNAi does not work. If you do the double RNAi, the root becomes less sensitive to ABA. So this is important now. If you overexpress this protein, the transgenic rice functions as if ABA is around or stress is around. If you knock down the expression of this protein with the RNAi, then this rice plant does not respond to ABA as the non-transgenic wild type. And finally, we notice you cannot just measure elongation because the lateral expansion is also affected. While the presence of this protein inhibits elongation, it actually promotes the natural expansion, so making the root shorter and fatter. So finally, back to cell biology. Where in the root is this protein expressed? So here we have the control. So we have the promoter of this gene linked to GUS and stand for GUS. In the control, that is, there's no stress, there's no ABA treatment. You see low level of expression of this protein throughout the root. But if you treat the root, transgenic root, with ABA or stress, you see those tiny dark blue spots. If you use lower magnification, if you use higher magnification, you see the blue region is concentrated in the elongation region. The other region, actually, you don't see a whole lot of expression. So here's the working model. Let me summarize it. We have discovered a monocot, unique, root-specific, highly protein-rich glycoprotein, and this is stress and ABA-induced. This protein is heavily glycosylated and also has the lectin activity. That is, it binds to a particular cell wall hemicellulose that's arabinogalactan, and we know some of the hemicellulose are important for cell expansion. And this protein is localized on the plasma membrane primarily. So what we believe is this protein localized on the plasma membrane, allowing it to interact with a very important component of the cell wall hemicellulose that is responsible for cell expansion. So the consequence is the cell wall cannot be expanded and cell elongation cannot take place. So that's the working model. Of course, we still have a lot to do. We need to understand the mechanism. We need to really figure out how this protein is targeted to the plasma membrane. So before closing, let me acknowledge the people who actually did the work. Yi Jie is the graduate student in Academia Sinica who has been doing 90% of the work. Tuan-hua Hong, who is also present in the meeting here, a former postdoc now, is a professor in National Taiwan University. He was responsible for discovering this protein in the first place. And we've been collaborating with Su-mei Yu, my colleague in Academia Sinica, for many years. Thank you very much. I guess probably I have time for one or two questions, if you care to ask. Jerry?
David, I guess in leaves, doesn't ABA sort of inhibit leaf expansion as well?
Yeah.
I wondered if this protein is working in leaves or if it's just in roots.
This is an important question, Jerry. We cannot really rule out the possibility that this protein is not important in leaves. But we think the relative importance is less in leaves. First, it's not really expressed at a very high level in leaves. First. Second, the transgenic plant overexpressing or with the expression level knocked down do not seem to have a very observable phenotype in the leaves. So maybe this protein is doing a small job in leaves compared to roots. Okay, if no question, I'll shift my hat. I'm going to serve as chairman of the session since I don't have the information here. I'll just see what I memorize it. The next speaker is Dr. Anne Divar from CNRS in France. She's going to talk about, I think, functional genomics root development program. Let's welcome Dr. Divar.